Assalamu alaikum. Today's discussion is on coronavirus. Coronavirus is a family of RNA viruses with cone-like envelope and spherical structure. This family of viruses has vast capacity for adaptation to new environment, transmission infection, and emergence of new diseases. Coronavirus can cause acute mild respiratory infection along with fever and cold. In this presentation, main focus will remain on SARS coronavirus and SARS coronavirus 2. The novel coronavirus that first emerged in December 2019 was labeled as 2019 novel coronavirus, China coronavirus, and Wuhan coronavirus in various medical journals. The provisional terminology based on geographical origin of epidemic was unpleasant and it can cause bias against a nation. So according to guidelines of World Health Organization, International Committee on Taxonomy of Viruses has given a neutral and generic name SARS coronavirus 2 to coronavirus and this name was given due to its resemblance with previously known SARS coronavirus. There is no any strong evidence that originally from where this highly infectious virus came into zero patient. Novel coronavirus is genetically modified virus and second phase mutation is observed in it. Being RNA virus, mutation in coronavirus is a natural phenomenon. Flight data is being used to generate models of virus possible flow around the globe. SARS coronavirus and SARS coronavirus 2. Both coronaviruses are spherical enveloped particles with positive sense single-stranded RNA genome. The spike proteins, envelope proteins, and membrane proteins are surface proteins present on the surface of virus particles. These surface proteins usually embed in lipid bilayer of host cell in case of infection. Nucleocapsid protein are also associated with viral RNA. This diagram of SARS coronavirus 2 has spike proteins and these spike proteins are associated with infection. Molecular study of these spike proteins reveal that they have 10 to 20 times more affinity for their receptor in host cell as compared to SARS coronavirus. That is why SARS coronavirus 2 is highly infectious. Now we will discuss about transmission of these viruses in humans. Transmission of these coronaviruses mainly occur through infected droplets and their deposition on respiratory mucosal epithelium. Bats are considered as potential intermediate host for the transmission of these viruses in host. Chicken, reptile are also considered as intermediate host. SARS coronavirus were also detected in tears of infected individual. Clinical symptoms of SARS coronavirus 2 are fever, influenza, and pneumonia. While in case of SARS coronavirus, decreased platelets count and mildly elevated serum hepatic enzyme. These two parameters were also observed in case of SARS coronavirus. SARS coronavirus and SARS coronavirus 2. Both coronaviruses share same receptor and that receptor is metallopeptidase ACE2. This receptor is angiotensin converting enzyme 2 and this is necessary for angiotensin production in host cell. Spike proteins of viruses attached with receptor ACE2 and the proteases, cellular proteases prime these spike proteins. These receptors and cellular proteases are necessary for infection with SARS coronavirus 2. Clinically approved protease inhibitors are available to block entry of SARS coronavirus 2. Viral spike proteins bind with receptor and conformational changes occur in spike proteins. These conformational changes facilitate the fusion of viral envelope with cellular membrane, and the virus enter by endosomal pathway into host cell. Then virus releases its RNA into host cell. That RNA is translated into viral replicase proteins. Further transcription and translation of viral RNA provides viral proteins. Genomic RNA and viral proteins assemble into virions in modified endoplasmic reticulum and Golgi complex. These virions then released into lumen of endoplasmic reticulum Golgi intermediate compartments in the form of vesicles, and these vesicles then released out of the cells. Continuous replication and expression of viral RNA genome is compulsory for onset of clinical symptoms of relevant disease in host. Now we will discuss about diagnosis of SARS coronavirus 2. World Health Organization advises to take sample from nasal secretions and throat with help of swab. For the diagnosis of SARS coronavirus 2, both ELISA kits and real-time PCR-based kits can be utilized. In case of ELISA method, antigens for viral proteins and antibodies that detects spike proteins of virus have been developed. These two things are used to develop ELISA-based kits. In case of ELISA, antibodies react with spike proteins of coronavirus, and sandwich ELISA provides 92 to 93% sensitivity and 100% specificity. ELISA provides positive result in case of low viral load in sample. PCR false negative can also be detected by ELISA. ELISA gives faster results as compared to PCR. Real-time PCR-based kits can also be utilized for diagnostic purpose. Real-time PCR usually detects presence of virus E gene. This E gene codes for envelope protein and gene for enzyme RNA-dependent RNA polymerase. There are also some other PCR-based kits available. These kits are monoplex and multiplex. Monoplex this determine S gene, while multiplex determines N gene and ORF1ab gene. Multiplex identifies the sequence published by Chinese Center for Disease Control and Prevention. All other things are similar in both kits. After positive results of test screening, whole viral genome screening is done for confirmatory tests. CRISPR technology can also be used to target viral sequence with fluorescence probe for the diagnostic purpose. Now we will discuss about treatment. As there is no any potential antiviral drug available, so physicians are using the symptoms to treat the patients. 80 different trials are going on in China for discovering potential therapy against SARS coronavirus 2. These trials include chloroquine and HIV drug trials on animal models. However, a California-based biotech company has made a drug which is a nucleotide analog. This nucleotide analog can block viral proteins and polymerases, so in return it can block replication of viruses in host. So there is a hope in this nucleotide analog. So now we are turning towards precaution. Personal hygiene, hand sanitization, observance of quarantine, and use of well-cooked food are best found precautionary measures. So Reverse genetics was found helpful to know about SARS coronavirus. Now, it again can be helpful to obtain information relevant to determination of virus growth in culture, virus transmission adaptation mechanism, host immune interference with viral replication, virus virulence and pathogenesis, virus genome recombination and mutation mechanisms. Investigation of molecular basis of infection and replication of SARS coronavirus 2 can provide information relevant to potential drug targets that may be helpful to stop any associated epidemic in future.